Hey, this is Dr. Ben Weitz, host of the Rational Wellness Podcast. I talk to the leading health and nutrition experts and researchers in the field to bring you the latest in cutting-edge health information. Subscribe to the Rational Wellness Podcast for weekly updates. And to learn more, check out my website, drweitz.com. Thanks for joining me, and let's jump into the podcast. So today our topic is the microbiome, which we all know is of major importance to our health. It's no accident that many of us in the functional medicine world tend to start by looking to the gut as the most important aspect of our underlying factor in many aspects of our health. Just to be clear, the more than 1 trillion bacteria, fungi, protozoa, and viruses that inhabit our digestive tract, especially in our colon, but also in our small intestine, as Dr. Pimentel explained to us last month, also on our skin, in the vagina, and on nearly every mucous membrane in our body is referred to as the microbiota. And the microbiome is actually the genetic material of all these microbes. The bacteria in the microbiome are incredibly important for our health. They help us to digest our food, regulate our immune system, protect against other bacteria that cause disease, and produce various vitamins, amino acids, short-chain fatty acids, and neurotransmitters, among many other functions. The father of functional medicine, Dr. Jeffrey Bland, once said, "The relationship between our diet and the trillions of bacteria in our intestinal tract is one of the most important features that regulates the way our genes are expressed." We now know that the concept of trying to map out the microbiome so that we can determine an ideal set of specific microbes that's going to determine a healthy microbiome is really no longer a practical definition. So we want to look into what factors can help us determine if a given patient has a healthy microbiome, or if their microbiome may be contributing to their disease or set of health conditions. For example, there's a growing body of evidence. There's a growing body of evidence showing that a more diverse microbiome is associated with improved health, and that a lack of diversity is associated with obesity, inflammatory bowel disease, diabetes, among other conditions. So I'd now like to introduce Tanya Elliott of Microbiome Labs, who's the sponsor for this evening, and after that I'll introduce Kiran. So where is she?
Let me see if I can.
So I have to find you on the
I think I unmuted.
Unmute you.
Can you hear me?
Okay.
Yeah. Hi, everybody. Thank you so much for joining us. I'm really excited that this was able to happen on such short notice. My name is Tanya Elliott, and I am the Regional Account Manager at Microbiome Labs. I cover Central California, which is Long Beach, basically, to San Jose. And I just wanted to, offer, my support during this time. I know things have been, off with COVID, but I'm here as a resource to help support you and your patients. And wanted to mention 2 of our new things that we've come up with is we have social media kits that can really help with reaching out to your clients and supporting them and supporting their immune system. And we also have our Patient Direct. So a lot of you are maybe now just starting to go back into your office, but we do offer Patient Direct so that patients can use a special link that's associated with your account and go on and order for themselves and have it shipped directly to their house. And, we've decided that it was important to up the amount of commission on that, so we've increased that to 30%. So if anybody would like additional information on either the social media kits or Patient Direct, I'm always here as a resource, and Ben will share my information, and I'll pop it into the chat room as well. And we're also offering a show special tonight. So this is our pricing for every 12 bottles you buy of one product, you get 2 free and free shipping. So it's the best pricing that we offer, and I highly encourage you to take advantage of it. I have a high attendance sheet that Dr. Weitz will share the link to, and you can fill that out, and I'll be able to contact you. And again, I'll share my information as well in the chat room, so grab that. Again, Tanya Elliott with Microbiome Labs. Super happy to see you, Kiran, and you're looking golden and like you've gotten a lot of vitamin D recently. So.
Great to see you too. It's been a long time since I've seen your face.
Yes, so too long. So. Thank you all for joining.
Thank you.
Kiran Krishnan is a research microbiologist and has been involved in the dietary supplement and nutrition market for the past 20 years, including hands-on involvement in university research. Kiran is a co-founder and the chief scientific officer at Microbiome Labs, a leader in microbiome and probiotic research. He's a frequent lecturer on the human microbiome at medical and nutrition conferences. He's currently involved in over 18 novel human clinical trials on probiotics and the human microbiome. Kiran is also on the scientific advisory board for 7 other companies in the industry, and he's published clinical trials in peer-reviewed scientific journals and several global patents in his name. Kiran, thank you so much for joining us, and especially that we were able to do this on short notice when the other person who was supposed to be speaking tonight kind of flaked out at the last minute a few weeks ago. So thank you.
Yeah,
thank you.
It's my pleasure. Thank you for having me. It looks like a great group, and I'm excited to have a good conversation.
Good. So what are the most important components to a healthy microbiome?
Yeah. So there are lots of little kind of fine details within the microbiome that can be critical. But as we've now exceeded, I think, around 60,000 publications on the microbiome, and that's just in the last 5 to 6 years, right? So it's one of the most active areas of research ever in the history of biological science. Over the next few years, we're going to have more papers published on the microbiome than any other topic in all of science. So it's a very exciting time. And the thing is, once we get to that point, we'll still kind of be at the tip of the iceberg, which is the crazy part about all this, right? So one of the underlying themes, and you mentioned this,
like when you say that, I mean, we've really only scraped the surface in understanding some of the different organisms that are present in a normal microbiome, right?
Exactly. Yeah. Number one, finding the organisms. Number two, how do they work with each other? One almost mind-boggling kind of example is just looking at the lung microbiome, right? So there's all this evidence that depending on what your lung microbiota looks like, you have different susceptibility to things like respiratory viruses, including influenza, COVID-19, and so on. This has been decently well studied in influenza, where when a virus enters your lungs, the microbiota picks up the signal that a virus has entered that space. The microbiome in the lungs then sends a message to the microbiome in the gut. Then the microbiota in the gut activate and recruit immune cells to go to the lungs, right? So it's mind-boggling, but how do they communicate, right? What is that kind of light speed almost communication? How does a bacteria in the lung know to send a message to the bacteria in the gut? That part, those are the things that are really mind-boggling. But to give you a general idea of what is consistently the information about a healthy microbiome, it's the diverse microbiome, just like you mentioned in your introduction. The diversity of the microbiome dictates so many things. It dictates ultimately how long you will live. There's a number of longevity trials on diversity, and they show that people that have really good longevity, who have low risk for chronic illness in their 80s, 90s, and so on, tend to have the diversity in the microbiome of 30-year-olds. Those that start to rapidly lose diversity start to age really quickly. So diversity is absolutely paramount. The other part of it is the presence of these protective keystone strains. There are certain strains, and so far we've identified somewhere around 13 that could qualify as keystone strains. They're so important because they are directly correlated with disease conditions. Akkermansia, for example, eosinophila, inversely correlated with everything under the cardiometabolic syndrome spectrum, right? So one bacteria in your gut can protect you against almost 60 different diseases, which is under that cardiometabolic syndrome, Caelenbacter, Prosnitzi, everything under inflammatory bowel conditions, various forms of colitis, Crohn's, colorectal cancer, inversely correlated with those conditions, meaning the more of that bacteria, the more protected you are. So in general, if you can think about improving diversity, increasing those keystone strains, you're already making leaps and bounds of progress towards a healthy microbiome, and then overall a healthy life and longer life too with that.
What about, we've heard a lot about the Firmicutes Bacteroides ratio. Mm-hmm. Is that something that's really important? I think initially it seemed like it was super important, and now maybe it looks like it's not that important. What do you think?
Yeah, so when they were initially studying it, what they were looking at was the correlation between Firmicutes Bacteroides ratio and obesity. Right. So what they were finding in very early studies was that if you had a Firmicutes Bacteroides ratio where Firmicutes was higher than the average normal level of Firmicutes, then you tended to be overweight. So you tended to have metabolic dysfunction, and that automatically creates a problem because it says Firmicutes are bad, Bacteroides are good. The problem with that is that Firmicutes and Bacteroides are large phyla of bacteria, right? So these are groups that are massive, and there are hundreds and hundreds of strains that fit into both of those categories. And between the two of them, they account for over 90% of the microbes that live in your gut. So there's plenty of really good and important Firmicutes, and there's plenty of good and important Bacteroides, and plenty of problematic ones in both, right? So it became too general to say Firmicutes was a little bit higher than the typical average, then you would be overweight. But one of the aspects of that ratio that has actually held true throughout the most recent studies is that if you tended to have a high level of Firmicutes, if the ratio was off, meaning the ratio was over typically 1, meaning you're supposed to have more Bacteroides, and then Firmicutes is somewhere under a ratio of 1 compared to Bacteroides. If it was over 1, it means that you have a higher risk for vascular issues and chronic vascular issues. So things that are associated with obesity, but you don't necessarily have to be obese to have them. So cardiovascular disease, hypertension, those types of things. So that's still there. And for the most part, what they show is like a slight adjustment in those ratios can be profound, can have a profound change. That's part of where they think that all of the health benefits from a fiber diet comes in. Because when you increase your fiber, one of the things that occurs is Bacteroides start to go up. A lot of bacteria in that category eat fiber and resistant starches as their primary source.
Right. So in terms of the obesity, I think that there may be a number of organisms that might play a role. I know we treat a lot of patients for SIBO, and methane SIBO is caused by archaea, and they are also associated with obesity because they slow down the motility, and therefore you absorb more calories from your food.
Yeah, absolutely. And then if you think about anything that causes inflammation in the gut is going to drive obesity, right? Because ultimately, the biggest driver of obesity is endotoxemia. It's the translocation of LPS. That's one of the first things that occurs that starts screwing up sugar control, starts screwing up metabolic control, and the response of the brain to food coming into the gut. And we saw this in our first published study on leaky gut. It was interesting to find this. We were measuring it, but we did not expect to see any real data on it, and that's on the leptin portion of the study, right? So because we basically recruited people of normal body weight, so these weren't people who were technically overweight, who had BMIs that were too high. These are normal body weight people, and then per FDA standards, they were healthy normals, meaning they had no chronic illnesses, they didn't complain of any issues, they weren't managed for any disease. What we saw in the people that had a severe leaky gut reaction when we gave them food was that they also had a complete attenuation of the response from the brain to the presence of food, right? So the way we measured that is we measured the hunger hormone, Ghrelin. So we had them fast and fasted overnight, so about a 12-hour fast, They'd come into the lab early morning, and then we'd take the blood and we'll look at ghrelin levels, which is a hunger hormone. And as you would expect, ghrelin levels are high, right, because they've been fasting overnight. Then we give them a 2,000 or so calorie meal, and we measure the ghrelin level over the next 2, 3, 4 hours, and we see that the ghrelin levels barely drop, and then over a couple hour period, it starts to go back up again to the fasted level. And that is ghrelin, the beginning of ghrelin resistance, right? Because that means your brain is not recognizing that there's enough food in the gut and stop producing that hunger hormone, right? One of the big factors of obesity is overeating. And so that ghrelin response is really important. So what we saw then when we gave them the spores in that 30-day period to stop that leaky gut, that LPS endotoxemia, they would come in and the ghrelin levels would be up because they're fasted. We give them the meal, and then the ghrelin levels would drop 50% within the first couple hours. All right, so we're showing complete satiety. And so that prevents you from overeating. Now, here's the other part of that. The other side of the coin of that is ghrelin plays as a yin-yang with another hormone called leptin, right? And adiponectin is another one as well. But just talking about leptin is then the satiety hormone. As ghrelin levels go down, leptin levels are supposed to go up. One of the functions of leptin increasing is to start turning on metabolic processes in your body so that you start burning calories, start burning fat, and so on. So what we're doing when we keep ghrelin up and we lose that response to ghrelin, leptin stays low too. So not only do we overeat, we don't have this hormone coming up going, okay guys, we got plenty of energy, let's start metabolizing stuff. So it's a real troubling pattern. And we were really excited, but also surprised to see that in healthy weight individuals. So these people were being set up for obesity over the next several years. They happen to be young, right? The average age in that population was somewhere around 22 or 23. So their body could still resist that leptin resistance. They were still very active and so on. But that maintenance of that dysfunction over the next few years, then they're going to start putting on the weight.
So let's try to translate that into clinical practice. We have a patient comes in our office and they're obese. What are we going to look at in the microbiome that might be related to that obesity? And then what steps can we take to modify it?
Yeah, so a couple of things. If you were to do a really good whole genome sequencing microbiome test, like we have the Biome Effects, not only does it give you super accurate because it's using whole genome sequencing, super accurate reads on certain types of bacteria, but it's also going to give you functionality, right? So a couple of the key things that you would look for is Akkermansia levels first. Akkermansia levels are going to dictate the risk for ongoing obesity and metabolic dysfunction. If their Akkermansia levels are just low, or in many cases, we find undetectable levels of Akkermansia, meaning it's probably there, but it's at such low levels we couldn't pick it up. Then the focus would be, let's get the Akkermansia levels up, right? And oligosaccharides do a really good job of getting Akkermansia levels up. Like that's one of the main reasons why we developed that Mega Pre product, is because it has the specific oligosaccharides, especially from kiwi, that bring up Akkermansia levels dramatically. In our study that we published in August of last year, we showed in 3 weeks we increased Akkermansia counts almost 1,000-fold in a 3-week period. So that's one of the key things. The second thing that brings up Akkermansia levels is fasting. Fasting increases Akkermansia because Akkermansia likes the mucin layer, and it will eat away at the mucin layer when there's no food coming through. But the good thing about it eating away at the mucin layer is that when it does that, it turns on genes like the MUT gene, which stimulates the regeneration of the mucus layer. So that's a very important normal part of the repair of the mucus layer. So you're giving Akkermansia a chance to do that, but it also then allows Akkermansia's population to increase, which then protects you against obesity and metabolic dysfunction. Another thing that stimulates Akkermansia is polyphenol intake. So polyphenols from things like wine, of course you don't want to drink too much wine, that'll be counterproductive, but fruits and colored fruits and vegetables like berries and things, and dark chocolate, those kind of polyphenols actually feed Akkermansia as well. So that's one of the key things. The other thing you would look at is the Prevotella Bacteroides ratio, not Bacteroides.
Now let me stop you 1 second. I'm sure a lot in the audience are thinking, why don't we just give Akkermansia probiotics?
Yeah. So that's a very important question, and in fact, I know for a fact that lots and lots of companies have been working on Akkermansia probiotics for a decade or so. The problem is Akkermansia is the single species in the genus Akkermansia, so Muciniphila is the species, and most of us have kind of our own unique version of Akkermansia. The other problem with Akkermansia, it's an obligate anaerobe, which means that even a slight exposure to oxygen will kill it. Right, so we're doing a bunch of work on anaerobic strains as well. One that we're working on right now is called Butyrate. Butyrate is one of the most potent butyrate producers in the gut. And Butyrate is an obligate anaerobe. It's a little stronger than Akkermansia, so if we work very carefully, there's a shot at making it an effective probiotic. But here's what we have to do for that. We had to build a facility, a production facility that's all anaerobic, meaning that whole building has no oxygen in it, right? So you're going to have all of this sophisticated machinery to suck all the oxygen out, air dampers that put in nitrogen into the facility, keep the atmospheric pressure at a certain level, and then all of the workers that are in there are in full spacesuits, basically.
Wow.
Right? So it's not an easy thing to do. And then the production has to be.
Like you just have the doctors, from the COVID ward just come right over.
Exactly. They're already geared up. Right? And here's the complexity of it is that we can't have any really major breaks in the production cycle, because anytime there's a stop and moving of material product to another area, you risk oxygen exposure. So the way the system is that we've been able to develop is you can actually start the fermentation on one end, and then it all becomes pretty much automated, and it comes out the other end in these highly specialized capsules that deliver it all the way down to the large intestine. Right, but it's complex, and the problem with Akkermansia is it's even more unstable than Butyrate. It's not ever designed to be outside of the body. But the beauty is we don't have to go through all of that difficulty, because we know how to grow Akkermansia in your gut, and your own Akkermansia is going to be more powerful than any other Akkermansia you might be able to implement. So that's a really important one. The other one is this.
Hang on 1 second. One of the questions just came in. What if the Akkermansia is too high? Isn't it related to certain autoimmune diseases like MS?
Yeah, so that is a rare problem. However, the elevated levels of Akkermansia causing problems seems to be, as they go deeper into those studies, seems to be also associated with having very low levels of Faecalibacterium prosnitzi. So there's a give and take there. If you have too much Akkermansia, you don't have enough Faecalibacteria, that may create a dysbiosis in itself. But in general, when you look at the studies, that's a really rare case. Most of the U.S. population tends to have very low levels of Akkermansia. And we've been running the BiomeFX test, right? We've got a few thousand tests already done from lots of our docs' patients, and we're getting this kind of data. We're seeing it, what are the trends that we're picking up among the U.S. population, the kind of people that are seeing functional medicine doctors, and we see a lot of people with undetectable levels of Akkermansia. So I think, there's an issue. Now, here's another thing with elevated Akkermansia. In general, if you tend to have a pretty diverse microbiome, a lot of those species kind of self-regulate, and they don't allow themselves to go above a certain threshold. And Akkermansia also tends to be concentrated in the distal part of the colon, right? So then you've got trace amounts of Akkermansia in like transverse and the ascending colon, but it mostly in the distal colon where you see a flourishing of its activity. So it knows it doesn't allow itself to overgrow in certain parts of the gut, but then it will have a certain level of predominance in the distal part of the gut. And part of that is because it does a really good job of metabolizing the food components that aren't digested in the rest of the intestine. Finally comes to Akkermansia at the end before it goes into the for defecation, then Akkermansia has a chance to work on it and metabolize it, and then send whatever it doesn't use into defecation.
So a question just came up, and it basically refers to what I'll call the probiotic paradox, which is we want to use specific organisms that we think will be helpful and reduce other specific organisms, but yet we know that probiotics are only temporary visitors. So even if you put Akkermansia in, they're not going to become permanent Akkermansia there. So does it really matter if you put a specific species in? But yet we know that there's specific benefits come from using specific species. So how is it that Akkermansia, which is not going to become a permanent visitor, even if we could produce it, but yet could have a beneficial effect for somebody who has low Akkermansia?
Now it could, it could, just based on its DNA being present in the system, right? That's how some probiotics function even without being alive. And in fact, there are several really well-known and well-studied strains like Rhamnosus GG is one example. Infantis 35624 is another example, where they have clinical studies on those strains, and they show clinical benefit, measurable clinical benefit of those strains. But then they've also done studies where they deliberately heat kill the strain and then administer the dead version, and in many cases the dead version works even better than the live version, right? So that is a paradox that has been created because the accepted scientific definition of a probiotic is a live microorganism. So if the organism is dead and still confers a benefit, then is it really a probiotic? And that's a bit of a debate in the whole probiotic microbiome world. The prevailing idea is that they are not necessarily probiotics, they are metabolic response modifiers, because they will tend to have, these specific strains will tend to have either DNA or RNA fragments, or certain types of peptides, or certain types of nanovesicles that they produce that can have a metabolic response in the body. Similar to how an herb would have it, or vitamin might have a metabolic response, they have something inside them that creates a metabolic response. And in part, that may be why heat treating them actually improves the effect, because when you heat treat a cell, you're basically lysing the cell, right? You're opening it up and you're getting all what we call the lysate out of the cell. There are lots of studies like in places like Russia where they focus predominantly on lysate, where they will take certain types of fermented and grown bacteria, they will purposefully kill it, and they're trying to pull the lysate out of the cells because they know that there's special compounds being produced in the bacteria during the fermentation process that you're not going to get out of the bacteria if you take it in. So they try to lyse the bacteria, pull out the lysate, and make that the therapeutic compound. So many probiotics on the market today work in that fashion. It's a temporary benefit as the bacteria and its components are moving through the system. It's giving you this metabolic response. Now, we started working with the spores because the spores play a little bit differently. The spores can absolutely survive through the gastric system, so they're not dying in the passage. And when they get into the intestines, they're actually functional in the intestines. But, like most people know, they don't stay forever. But what's interesting about the spores is their ability to change the rest of the microbes in the system, right? So when we first started looking at the microbiome and things that may be beneficial, we concluded that since you've got somewhere around 30, 40 trillion organisms in your gut, right, no matter what we put in there, even if it's a super high dose, 300, 400 billion, right, it's a tiny drop, a minuscule drop compared to the sea of microbes that are in your gut. So any effect that we hope to get, that we think would be profound, would have to come from that probiotic being able to influence the rest of the microbes.
Okay. So we know that we have all these bacteria that are an important part of our microbiome, but are protozoans and worms important components of a healthy microbiome?
Yeah, so there's certainly some populations that have more of those kind of organisms in their gut, depending on their culture, where they live, what they sleep and eat. But things like amoebas, parasites, those are all also normal part of the microbiomes. In fact, parasites can be extremely useful for immune function. There are parts of Europe where they use helminth and parasite therapy just to improve allergy response. Almost anything that can get in and stoke your immune system to a certain degree without actually causing infection is really beneficial to your overall function of the immune system, right? And most recently, I've been doing a lot of lectures on the role of the microbiome in immune response, especially when it comes to viral defense, because that's, of course, on the forefront of everyone's mind. So there's two claims that I make about the microbiome and the immune system, which we can dig in and explain if people want to know a little bit more about it. Claim number one is that the immune system would actually cease to exist in large part, in the way we know it, without the microbiome, right? And I can explain why that is. The second claim is that the microbiome is the neighborhood watch for the immune system. And that's a really, really important aspect of the microbiome. So I'm happy to jump in and explain those two things. If that's
Sure.
Yeah?
Yeah.
Okay.
Yeah.
Let me explain then the first claim about the immune system would cease to exist the way we know it without the microbiome. So most people know that most of the immune cells are made both in to some degree in the bone marrow and then in the thymus as well, right? So of course T cells made in the thymus,
Right.
hence they're called T cells. So when these immune cells are made in these different components of our physiology, they are then shuttled and transported to areas like lymph nodes, which is where they sit and wait for maturation. And in fact, they are pretty naive, and they really can't do much of anything until they get a certain degree of maturation. And when they get maturation, if they're needed, then the proliferation also occurs in these localized lymph nodes and in areas like the Peyer's patches in the gut. Now, the signals for the maturation and proliferation
come from the microbiome predominantly, right? So we have certain amount of machinery to make immune cells, but we're making baby naive immune cells, and it's the role of the microbiome to train, mature, and then proliferate those immune cells in order for them to actually do their job. The analogy I give is like, we can make children that are immature and small and tiny, and we can't send our 15-year-old without any equipment to war, right? If we want them to go out and defend the country, they have to go through basic training and all the training, get the equipment and all that to actually be able to go and fight. So what our bone marrow and what our thymus is doing is actually producing children, essentially, which are ineffective at fighting for us. And then our microbiome trains those soldiers. So without the function of the microbiome, you would not get maturation of those T cells, B cells, and macrophages, and so on. And this has been studied extensively in no-biotic mice, for example, the germ-free mice, right? When you rear a mouse germ-free, meaning it has no microbiome, you see very poor and complete attenuation of the development of the immune system. In fact, the amount of immune tissue in the gut, which is where about 70-80% of your immune tissue exists, that becomes attenuated dramatically. So you don't even develop all of that immune tissue in the gut. A second part to that first claim is a lot of the energetics that the roving immune system requires in order to continuously circulate through, and then when they find something, attack it, eat it up, use the complement system to neutralize it, or the natural killer cells and their ability to use nitric oxide and all the superoxides, all of that equipment and energetics come from the microbiome. So the microbiome produces the equipment for our defense cells to kill off viruses, bacteria, infected cells, and they produce the energy. Like butyrate is a really important energy source for macrophages and dendritic cells who are circulating around trying to protect us, right? So again, that relationship is so intimate, and the immune system would cease to exist and function if it wasn't for the microbiome. Then the claim about the neighborhood watch. I've been trying to illuminate the importance of the microbiome for people using what I think are sensible analogies. So think about inside the body, everything is covered by a mucosa. Right. So we used to say that the skin, the outer skin, was the largest organ in the body. It's a huge barrier. It's protecting us. But the skin is only about 2 square meters in surface area. The mucosa on the inside is about 400 square meters. Right. So it is way larger in terms of area than the skin. And most people here are from the US, so translation of 400 square meters is it's close to 4,000 square feet. So imagine an apartment or a house that's 4,000 square feet. We'd be pretty happy in that space. All of that is packed in as our mucosa, right? So a virus or bacteria basically enters through a mucosal layer, whether they're going in through the eyes, nose, mouth, they're going in through the urogenital tract, or even through the skin, they're going to enter the mucosa. So that's where all the sampling and action takes place. Now, in this mucosa, you've got about 40 trillion microbes that already live there. Right? So imagine you've got 40 trillion microbes covering this space where new pathogenic microbes enter, and it's the job of the immune system to somehow sift through all of the 40 trillion to find the 1 or 2 that might be causing you a problem. And in fact, to survey those 40 trillion or so microbes that already live in your system, you've got about 200 million immune cells that do that job, right? So it's a 200,000 to 1 ratio. It's an absolute mind-boggling feat when you think about it. The analogy I give is, imagine you're at a music festival, and it's good to imagining it because it's never going to happen again, or at least not for another year or more. But imagine we're at like a really fun music festival, and it's in a pretty big field, and there's 200,000 attendees there, right? So that's a lot of people. It's a big event. Among that 200,000 attendees, you get word that there may be 5 that are potentially egregious, that could be toxic and somehow hurt some of the other attendees, could bring all kinds of paraphernalia and drugs and really kind of cause issues. And you are the lone security guard in that sea of 200,000 people. And it's your job to find those 4 or 5 potentially harmful attendees, right? It would be an impossible task. The only way you could do it is if the other 199,995 attendees are also keeping eye for you, being a neighborhood watch, and can radio you should they see anything suspicious, right? That's the only way you can survey it. That's what occurs in your body. The sea of microbes that predominate cells, all of the cells in your body, they are the neighborhood watch for the immune system. When they sense a new pathogen entering a system, whether it's in the lung with your lung microbiome, or in your sinus cavities, in your gut, anywhere else in the body, the microbiota, the resident microbiota in that space, signals to the immune system that something is going on here. You got to come pay attention, right? That's the only way the immune system can survey this amazing surface area that is already covered with potentially harmful microbes.
Somebody asked the question, I guess you had mentioned the Prevotella Bacteroides ratio, and maybe you could explain what that is and its importance.
Sure, yeah. And what's interesting, another interesting thing we're seeing, and Prevotella and Bacteroides are also 2 phyla. And this is not Bacteroidetes, which is the one we were talking about with Firmicutes. This is Bacteroides, so it's very close in name but slightly different. What we're seeing is that when people consume lots of meat and protein, you tend to start to lose Prevotella, and then you start to increase Bacteroides. And again, not to confuse people, because this is not the Bacteroidetes we were talking about earlier. This is Bacteroides. And what we start to see in this pattern is the formation of insulin resistance, right? There's 2 things that occur. One is the inability to balance blood sugar levels. And then the second thing is the inability to build glycogen storage. So people that tend to have really low Prevotella and much higher Bacteroides tend to have blood sugar dysfunction, and then also tend to have get really lethargic and tired at some points of the day. They can't produce the energy. This is really profound because they're seeing this more and more in the Western world. And when we do the Biome Effects test, this is another one of those functionalities that we measure. What's interesting about when I've done a consult, so one of the things we offer for all of you guys that you should know, when you start getting the test done for either yourself or your patients, we offer consults with either our BiomeFX clinical director, Dr. Sam Molt, or we've got groups within our learning and development team that will go over the test with you so you get familiar with it, right? But for some practitioners who are friends of mine, I've done it myself, and they'll send me their patient's test beforehand, and I'll tell them, don't tell me anything about the patient. In the first few minutes I'm talking to you, I want to guess what they're coming in to see you about, just from looking at their test. And so far, I think it's been 5 out of 5 that I've been able to guess. And the last 3 have been this same issue. Like, are they having glucose metabolism issues, which they may not know because they're not necessarily pricking their finger and testing the glucose levels because they're not diabetic. But the way it cycles is it causes massive drops in blood sugar levels in the middle of the night. Some of these people awaken because of that. They get a panic response. During the day, they have real big swings in energy levels. And then when you encourage them to do a 24-hour glucose and insulin cycle, you see these massive swings up and down throughout the day. And they're not diabetic. Their A1Cs may be a little bit elevated, but they're not completely diabetic. And all of them have this diminished Prevotella and really high Bacteroides, right? So that's another function that dictates, like, and it doesn't have to be obese. Like the last 2 people I talked to about this, one was 110 pounds, so she's not in any way, shape, or form obese, and never has been. But because of multiple rounds of antibiotics and because she has SIBO-like condition, she's basically completely eliminated plant-based foods, and she's very high on the protein, and that starts to cause that dysfunction. So that's another one with that we tend to look at with people with metabolic dysfunction is how do we bring back that ratio, get that akkermansia up. Between those 2 things, you'll start to see a big change. We have a study publishing just on that recently. We took sorry to keep running on about this, but we have a study publishing recently where we took obese individuals. So these are people with a BMI of 30 to 31, somewhere around there. So they're not morbidly obese, but they're overweight. And we did full DEXA scans. We were looking at visceral fat, subcutaneous fat. We were looking at a whole bunch of metabolic parameters. It was a 90-day study, right? And what we did is all we added into their system, and this was a placebo-controlled study, was a little bit of a prebiotic that we know increases akkermansia and bifidobacteria as well, and then the spores, the spores that stop the LPS, the leaky gut. It was a 90-day study. We told them not to change anything about their diet. Don't add any exercise in that they weren't normally doing. So they are continuing all of the behaviors they normally continued that kept them overweight and kept them gaining weight, right? And what we saw in that 90-day period with no changes in diet, exercise, or anything was in the group that was getting that probiotic-prebiotic combination, we saw a 38% reduction in visceral fat mass. And that's the really dangerous fat around the organs, right? We saw some weight reduction as well, but weight is just not a great measure of metabolic activity. But doing the DEXA scan, we saw real fat being lost quite dramatically. And we also saw an inching up of their lean mass. So they were putting on some lean body mass. And then we saw all of these beneficial metabolic changes as well. So that just goes to show, without even diet and exercise and those other aspects of it, just shifting the microbiome, we can start shifting their metabolic profile. And then if you add diet, exercise, and all that to it, the results can be quite profound.
That's amazing. Can this be used as an adjunct to working with the patient with diabetes or pre-diabetes?
Yeah, absolutely, yeah. In fact, we're starting a pre-diabetes study with UCLA right now on the same principles. But in this case, we're going to have the probiotic. We're going to have components of the prebiotic, but we're also adding in bitters. In fact, berberine. And one of the important
natural metformin.
Exactly, completely natural metformin. And one of the things that's really interesting about it, and the professors that we're working with already have published on some of the mechanism of action of bitters. We have bitter receptors right in the upper part of our GI, right where the stomach dumps out, and then in the lower part of our intestines as well. Those bitters trigger things called peptide YY, GLP-1. These are transducers or transmitters that actually improve leptin response, increase fat burn by increasing something called cyclic AMP. So it stimulates all of the cells in your body to start burning fat for fuel. It improves gastric emptying and motility in the gut, and then it dramatically improves the insulin sensitivity as well. So simple thing like that, and and we know that. You guys all know that prediabetes is going to be one of the biggest health pandemics in the next couple of decades, because not only do we have so many diabetics right now in the world, we have 4 times as many pre-diabetics as we do diabetics. And almost 80% of them are going to become diabetics over the next 10 years. And when they all become diabetics, morbidity and mortality for all other chronic conditions increase dramatically with diabetes, including COVID, right? The mortality rate for diabetes with COVID is 7 times higher than a non-diabetic. And that's scary, because in the US, we have lots of adults and even some children who are diabetic.
Absolutely. Get into discussion with somebody about COVID, and they go, well, it's only people who have these high risk factors. And when you look at the American population, you're talking about obesity. We got 70% of people are overweight. You add in a number of people with diabetes and heart disease and autoimmune or immunological compromise, and then you add some cardiovascular conditions. You're talking about a huge percentage of our unhealthy population.
Per CDC data, 60% of US adults have at least one of those chronic illnesses. Falls under the high-risk category, right? Hypertension, diabetes, cardiovascular disease, obesity, 60% or more. So we have a tremendously high-risk population in general, which goes beyond just the age factor, right?
Right, and this should be one of the messages we get out of this pandemic crisis we're in, is we have a really unhealthy society, and we've got to do something about improving our overall health.
Yeah, absolutely. And that to me was, I've done lots of interviews so far on COVID and the pathology and so on. One of the first interview I ever did, I said, my point was that there is a silver lining here, because when you look at this virus, it's developed the capability of taking advantage of our vulnerabilities. And that vulnerability is the ACE2 receptor. And the ACE2 receptor is expressed in cases of chronic low-grade inflammation, which is an epidemic in our society. And this pathogen has figured out a way to take advantage of that vulnerability. What it tells us is that we should have resilience against these kind of things, and many people do. But it's really painting a picture of how vulnerable we are as a population, because the thing is, we'll get through this one, right? And it's not as bad as it could be, as other viruses could be. It doesn't have the mortality rate as MERS did, or the first SARS did, or Ebola does, right? It doesn't have 15-16% mortality rate. But somewhere around the corner, there's another one of these coming around, right? And it could be as infectious, but 10 times the mortality rate, again, taking advantage of our vulnerabilities. So to me, that is really the silver lining in all of this, is like, we should realize that as a population, we are ill-equipped to handle something like this, only because our systems are vulnerable. Our immune system can take care of this kind of virus very easily, if we are allowing it to function in the right way.
So bottom line, for patients who are overweight, a couple of people ask questions about this. What can we do now to help in terms of the microbiome to make it easier to lose weight?
Yeah. So really important practical things. And this is exactly what I do too. And I'll go through swings with my own weight, and I actually just saw my sister the other day, and she's like, how did you lose? I leaned down over the last 2.5 weeks because I was like, I'm getting a little too pandemic chubby, so I got to lean down. So what do I focus on when I do that? And what I think will be really effective to start stoking people's metabolism. Number one is the fasting, right? If you can add in some intermittent fasting, and yes, it'll be hard for certain people to go 14, 15, 16 hours right off the bat, but you can totally taper them up, right? If they can go 12 hours without eating, 8 p.m. to 8 a.m. for the first week, have them do that. Next week, go 8 p.m. to 9 a.m. Start pushing them, right? If they can get to that 14, 15-hour range, metabolic changes will already start to happen. Akkermansia levels will increase. The microbiome diversity will increase. Bifidobacteria levels will start to increase. Those will be profound changes for their microbiome.
So why will our microbiome flourish when we don't eat anything?
Yeah, so that's a really important question. So part of it is about food utilization, right? So if you think about it, every meal that you eat takes about, depending on your system, but it'll take 8 to 12 hours before you defecate that part of the meal, right? It takes that long to go through your system. Now, the way the microbiome is organized is there are many community structures that feed off of one another. There are primary digesters of the macronutrients that you take in, and then they produce secondary metabolites. And then those secondary metabolites are metabolized by another group of bacteria that then produce tertiary metabolites. And then another group of bacteria can eat those and produce another set of metabolites. So that's part of the reason it takes so long, especially when the food gets to the colon, for it to start passing through and all of the digestion that occurs in the colon to happen, or at least the fermentation to happen. And the thing is, when you have primary digesters, let's say bacteria A is a primary digester of the carbohydrates that enter in as food products, right? Bacteria A sees the carbohydrates, it becomes really active, and it starts digesting those carbohydrates. When it is active, it suppresses the growth and the functionality of bacteria B, C, and D. Right? So it starts metabolizing, and then it produces all of these secondary metabolites. Its primary food is gone, so it starts lowering its activity. Then bacteria B jumps on these metabolites and starts producing. Bacteria B gets to flourish to a certain degree. When its primary food source is gone, it goes down the row, bacteria C starts to flourish. So when you give your body time to digest the food and ferment the food completely, you actually get to flourish lots of groups of bacteria. What tends to happen is if bacteria A is digesting its primary food, right, and then that food is gone, it kind of takes a metabolic rest, bacteria B starts to digest the metabolites, all of a sudden the primary food comes back in, then bacteria A start getting active again, which suppresses this next group of bacteria, right? So it's this staircase type of metabolic process. Hopefully I know it's a little abstract to think about, but hopefully that is understandable. So when you give your gut time to actually process all of the food, you get complete fermentation and digestion, and that completeness in fermentation and digestion allows more and more microbes to flourish that utilize different parts of the meal and diet. So that's a really important thing to understand. So if we just continuously keep feeding, we're really only supporting certain categories of bacteria. Others get suppressed. They don't ever have a chance to flourish.
Would it be even better to fast for 3 or 5 days?
So there can be some diminishing returns if you go too far. And there we need more studies on that. There's a lot of great studies on intermittent fasting. There's some pretty good studies on 24, 48-hour fastings. But there's very few studies so far on longer fasts, like 6, 7, 8 days, right? I would think that there's a point in most people's microbiome where you start losing the benefit and start gaining some dysfunction from that longer of fast. It's probably less typical for us as a species to go that long without eating than it is to go 14, 15, 16 hours, right? Our ancestors routinely went that long without eating because they didn't always have food at the ready. They would wait for the hunger pangs to kick in to motivate them to go out and start looking for food. So I would say you push that. The longest I've ever done is about 30 hours. And I could tell for me, and I intermittent fast every day pretty much and have been for the last 3-4 years. That 30-hour period was I could feel was not really producing much more benefit for me. So then I had small meals. I started feeling great. So that intermittent fasting becomes really important. That's one of the easiest things to implement. If you could start getting some prebiotics into them, the oligosaccharides in particular, the ones that we use is in that Mega Pre, the fructooligosaccharides that come from kiwi especially are really important for increasing Akkermansia and increasing bifidobacteria. Bifidobacteria is another group of bacteria that are inversely correlated with metabolic dysfunction. Now, the question is, if I take high doses of bifidobacteria, will I achieve the same thing? In fact, you don't. You don't see studies that show high dose of bifidobacteria having metabolic impact. There is one bifidobacteria strain called BLP1, I think, that when it's dead, will actually help induce some metabolic improvements. That's, again, one of those special cases where there's something within that bacteria strain that when it's killed and it opens up will actually provide a metabolic benefit, right? So the prebiotic will do a lot more to increasing your endogenous bifidobacteria, giving that metabolic support. Using the spores can be very powerful, the megaspore, because it stops that leaky gut. So you want to stop the leaky gut. You want to use a prebiotic to increase Akkermansia and bifidobacteria. You want to add in a degree of fasting. And then if you can get them to take additional things like resistant starches, that'll help, because one of the really important metabolites in the microbiome that controls body weight, controls metabolism, are short-chain fatty acids. Right? Butyrate is so important for metabolic health. Butyrate is one of the main signals that turns on the cyclic AMP process that causes all of your cells in your body to burn fat. Butyrate also helps with the leptin response and the satiety signaling. So they're really, really important. If you can just increase short-chain fatty acids, you'll start to see changes in weight.
Is it beneficial to take supplements of butyrate and short-chain fatty acids?
Yeah, that's an interesting component of butyrate therapy, because what they found is that butyrate is really useful in things like colorectal cancer or really severe colitis, right? When you have inflammation in the large bowel. But when you take it orally, it doesn't seem to help. So what they've started doing in hospitals is they'll do butyrate enemas instead to get it into the large bowel so it actually has that function. So there's a big question of whether or not orally supplemented butyrate actually will get to the large bowel where it needs to be, or does it get dissipated and absorbed in the small intestine, or maybe even the stomach, right? So that's the part that's unsure. Increasing your endogenous butyrate is not hard at all. Our study that we published last year showed that when we added the prebiotic and the probiotic in, we increased butyrate production by 150%.
One of the tricky things about using prebiotics is a lot of us have patients with SIBO, and they're very sensitive to prebiotics and fiber, and it tends to flare them up.
Yeah, that is problematic. So we say with the prebiotic that we use, which are highly specialized oligosaccharides designed to make it to the large bowel, so they should not have a lot of bacteria in the small bowel that can ferment it to create the gas and distension. But we tell them to test it. They can go as little as like 1/8 of a scoop. Mix it into a jug of water, shake it up, and kind of sip it throughout the day. We've even had some people that go as far as, not being able to take it in orally, so then they do a prebiotic enema to get some oligosaccharides into the large intestine, which is where the bifidobacteria, Akkermansia, all are there. So you can do a retention enema with the prebiotic mix. So that's one way to start getting it in there, if you can, because if you can't do it this way, you might try the other route.
So it's interesting that some of these probiotics that are dead are actually more beneficial. Myself, and I'm sure a lot of the practitioners who are listening, probably have probiotics in our refrigerators at our office, which we've made a big deal out of, making sure that they were refrigerated the whole time, thinking that we're providing an extra benefit because, if the temperature gets too high, the bacteria will die, and these are more potent. Are we really helping our patients?
Yeah, no, all of that work to try to keep it alive in the room is really all for nothing, because the moment they hit 98.6 degrees in the body and pH of 1.5 or something going through the stomach, they're just obliterated. And we've tested that. We've tested 40 or so of the top retail probiotics and even the probiotics in the health space, and all of them die going through the gastric system. So the key is then finding ones that have shown studies that the dead versions are actually beneficial, right? And that's specialized, right? Like, for example, with Rhamnosus GG, when you kill it and you administer it, it has all of these benefits, but regular Rhamnosus, the generic version of Rhamnosus, wouldn't have that same effect. You kill it, it's just dead and it's not really doing anything. You're going to poop it out 12 hours later. So the metabolic response modifiers, the types of strains that when dead can have specific metabolic effect, are very specialized to those subspecies of the strain. It doesn't translate to the wild type version of the strain.
With respect to intermittent fasting, somebody asked a question. Do you think it's more beneficial to skip breakfast or to skip dinner?
Great
you
question.
Think it matters?
Yeah. So yes, there are at least 2 studies that came out that showed that if your fasting window is basically from like from the afternoon till the morning, that actually has a bigger impact on weight loss than the overnight to the afternoon fast, right? So if your fast was somewhere around like 12 or 1 p.m. all the way till like 8 or 9 a.m., that seemed to have a more profound effect on weight loss and fat loss than the overnight to noon fast. Now, that being said, the overnight to noon fast also has benefit, right? The problem I have, and I can't do the other way around, just because dinner becomes such a big, important social thing, right? Whether it's business related, it's family, it's friends. So it becomes really hard to skip dinner. It just from the social side of it. And so that's why for me, I've not been able to do that kind of routine. I can do the skip the breakfast very easily. Now, the other advantage for me in skipping breakfast is that I can do my workouts in the morning, and doing your workout in a fasted state really amplifies your growth hormone levels, and that has a huge metabolic impact as well. If you can fast through the late afternoon and evening, and then throw in a workout somewhere in that evening period, that's fantastic. But a lot of people find it very hard to skip dinner. It's just there are too many social things surrounding dinner.
Yeah, actually, for me, it's easier because a lot of times I'm busy with patients and I don't really have time. And a lot of patients want to come in after work. And so rather than eat late, a lot of times I'll just skip dinner just because it's easier.
Yeah. That actually has a bigger impact metabolically than skipping breakfast. But if you can't skip dinner, then skipping breakfast will help as well.
Is there ever going to be a probiotic protozoa or fungus?
Fungus, yes. So we already have Saccharomyces, right? So
yeah, that's true. That's
well
true.
Established. We will have, I believe we will. We'll have potentially things like amoebas. We'll potentially have protozoas. We may have organisms that we don't even know exist yet. When prions became a thing with mad cow disease 20 years ago, I was actually still in university, and it was so interesting to see my immunology, microbiology professors like losing their minds as to what the hell is this weird alien thing, right? Because imagine viruses are really fascinating all themselves because they're not even living creatures, right? Viruses are technically not alive because they cannot create any sort of metabolic effect on their own. They can't reproduce on their own. They're just like a fatty envelope with RNA or DNA sitting in it, or a protein envelope with RNA and DNA sitting in it and just floating around, right? And then it just has this mechanism that allows it to bind to certain things, inject its RNA or DNA, and hijack a cell. So that in itself is fascinating. Then you think about a prion, which doesn't even have that sophistication. It's just a strand of protein. And this strand of protein, it just so happens the prions that we know of right now are really devastating. That's what created the mad cow disease, right? So it creates the encephalitis. So it basically busts holes in your brain. But it's this tiny little thing of protein, and there's like no functional way of killing it that anyone knows of. It can withstand all kinds of heat. It can withstand all kinds of acid. It's really mind-boggling. So we may find organisms that we don't even know exist right now that are really important therapeutics within our system. I think the next ones beyond bacteria and fungus, are likely going to be in the parasite family, in the parasitic family, just because there's so much work being done with that already in a fringe world.
Right.
The Helmite therapy and all is considered by many to be like really weird, but the data is good, and they're showing real progress in certain parts of the world in treating allergies. And when something like allergies becomes more and more of an epidemic in our country, we'll start having to go towards those types of therapies.
Right,
Now,
and don't we know that societies where worms are endemic have much lower rates of allergies and asthma and autoimmune diseases?
Yep, absolutely. And there's a double-edged sword that there are certainly some worms that will then create all kinds of other problems down the road, but absolutely, and I think in part what that speaks to me is it speaks to our disconnect from the natural world, right? So we've evolved to have a pretty intimate relationship with all of these organisms in the natural world. And the more we disconnect from them, then the more susceptible we become, because we're missing key links to our function and our phylogenetic tree. And so understanding what those things that we're missing are and putting it back into our system, I think will be a really big part of microbiome therapeutics moving forward. We are engaged with lots of very big companies in the world, and massive companies. What's exciting about it, and many people are turned off by massive companies, I totally understand that, but massive companies like Nestle, for example, who've spent their whole lives feeding sugar to people and sugar to kids, when you look at their new function, their new focus, they're saying we're going to de-invest from all of our candy, sugar stuff and focus on health, wellness, and microbiome science, right? There are companies like Chris Hansen and ADM, Archer Daniel Midland. These are massive billion-dollar companies, and they're all seeing the writing on the wall. I get calls from these kind of companies, and I talk to the leadership within those companies. They're all trying to figure out, how do we focus and hone in on the microbiome, right? They all see the microbiome as like the big thing over the next several decades to really improve health and humanity. So it's really exciting. I think we'll see lots and lots of innovations. One of the areas that we'll start to see more, and I'm always advising companies to this respect, is the microbiome modulation. Right, we're getting to understand more that if you have high risk for certain types of cancers, you have certain type of microbiome. There are certain features within your microbiome that are characteristic of that condition. So as we can better monitor and understand and test for those features, we can hopefully modulate features as we go along to fix people's ecosystems in a very precise way to improve their overall outcomes.
How does the microbiome change with diet? You mentioned that if we eat a heavy meat diet, that'll influence the diet one way. And right now in the diet world, we have these really extremes. We have people eating meat only, the carnivore diet. We have people only eating vegetables and everything in between.
Yeah, absolutely. And that to me is one of the scary things in the whole world of wellness and functional medicine, is the adaptation of real extreme and exclusive diets. We're just not designed to function that way, right? I think one of the things that makes the human species really unique in the animal kingdom is our incorporation of a really large diversity of microbes into our system, which really gives us strong omnivore qualities. And that omnivore quality actually is a big part of our evolutionary ascent up the ladder. Because if you think about other species that we would be competing with that are obligate carnivores or obligate herbivores, they are very susceptible to environmental and other changes, right? You think of a lion, as powerful and ferocious as a lion is, if there's a drought and the wildebeest levels are low, or its main prey, the lion's going to starve to death. right? If a human is in that same drought, we can go and dig for roots and tubers. We can eat insects. We can eat plants. We can eat rodents. We can eat a whole bunch of different things and survive through that. The same with on the opposite end with the lion's prey, the wildebeest, who's an obligate obligate herbivore. Can only eat plants. And so if those plants are dead and the savanna is dry, they're going to die, right? They can't just go and eat insects, and they can't just go and catch a muskrat and eat it. So we've got this really unique capability of producing tools using these hands. We're upright, we're bipedal, we can move long distances, and we can eat lots of different things. Our microbiomes are really designed to do that. So the moment we go to a very exclusive diet, especially if we do it for a long period of time, it's going to have a long-term negative consequence. I think short-term, like if you're really trying to change your metabolism and you're going to go keto for a short period of time, that could be totally fine, because one of the big benefits of keto is you're actually eliminating sugar. That to me is where you get a lot of the benefits of keto. So you can go keto for a short period of time, but then get those vegetables and the plant-based foods back into your diet. You also shouldn't be going vegan forever, and not getting any complexity in your amino acid profile and so on. So to me, it's really about a much more of a balanced diet, and I think that's how we are evolved to exist.
It's a really big current trend in the functional medicine world, as you mentioned, to talk about the dangers of eating lectins, the dangers of eating meat, because everybody's going to get heart diseases. And it seems like everybody's looking to the specialized diet that subtracts certain types of foods as the answer to fixing people with certain types of problems.
Totally, yeah. And that's a problem because, what we're not talking about enough is how do you build a resilient microbiome that handles all of these things, right? I mean, you talk about lectins. One of the types of diets that tend to have a lot of lectins is the Mediterranean diet, right? They eat lots of tomatoes and all that. It's one of the healthiest diets ever. There's more studies on the Mediterranean diet than any other kind of diet. And we're talking huge studies. We're talking 50,000 patients, longevity studies on it. So clearly it has benefit. And there's a way of eating it, and there's a way of preparing it. And the people's microbiomes are resilient to handle it. We have amazing capability of neutralizing things that are not actually good for us, the anti-nutrients people talk about. If we have a resilient, diverse microbiome, we can absolutely neutralize those. We can neutralize glyphosate in our gut. Not to say that we should have glyphosate in our food, but just to show the ability, the resilience of our microbiome to protect us. So to me, and people always ask me, like, what is your focus and goal with your microbiome? What do you try to do? And to me, my goal for health is about being resilient. Because I don't want to have to live a really structured, control life in order to feel good, right? Like you talk to a lot of people and go, how are you feeling? How's your gut doing? Oh, I feel great as long as I don't eat this, and they rattle off 17 things, right? And I feel great as long as I don't do this.
That's
right?
One of the biggest issues of a lot of us in the functional medicine world. We have these patients that come to see us for these severe gut problems, and gluten was a problem. They cut out gluten, they cut out dairy, they cut out this vegetable, they cut out that vegetable, they cut out, they went on a low FODMAP diet, so they cut out all the cruciferous vegetables, and then they found out about the next category of foods to cut out, and they come in and they're eating 2 or 3 fruits, and they're afraid to eat anything, and their gut has no tolerance to be able to eat these different types of foods.
And the thing is, the elimination doesn't do the healing, right? And that's the important part that people need to understand. In fact, speaking of gluten, there's a really good study on this, and I think it's fine for people to avoid gluten if they want to reduce the risk for that permeability. But you cannot avoid carbohydrates or cannot avoid fiber, because one of the big studies on gluten, they showed that in the first year of gluten intolerant people going on a gluten-free diet, their mortality rate almost doubles, right? And you would think like, wait a minute, that makes no sense. So these are gluten intolerant people, meaning when they eat gluten, they get all this inflammation, they get all this intestinal permeability. Why is it when they go on a gluten-free diet, does their mortality rate increase in that first year, and then it tapers off a little bit in the second year, but in that first 24 months, their mortality rate almost doubles, right? So the reason for that is in Western society, in our culture, in our society, more than 85% of our fiber intake comes from wheat, right? So you're getting all of this fiber despite getting the gluten that's also harming you, but you are getting the fiber component. So then all of a sudden when you eliminate gluten, and most people who eliminate gluten will replace it with proteins and fats, right? So you're taking out fiber completely out of your diet, and then you're replacing it with fats and proteins. So now the expectation is, well, I remove the offending thing, my gut should be healing. As it turns out, when they look at these individuals, the gut doesn't heal at all, right? It stays susceptible. And because you increase the intake of other things like fats that can increase endotoxemia, you are actually increasing your risk for other conditions. So what you should do in that case is eliminate the gluten is fine, but you have to replace it with other non-gluten sources of fiber and so on, because that is what drives the repair. Just eliminate is not going to cause repair. We need to maybe temporarily eliminate foods that are clearly offending your system, but then, make sure we're getting things in there to provide some diversity, provide some resistant starches, some fiber. All of those things are really important.
Is there any way that people following a carnivore diet, by eating the entire animal, every part of it, is there any way they can have a healthy microbiome?
So if they're eating the entire animal, then potentially, and that would include intestinal content. And we see that in hunter-gatherer tribes, right? So in the Hadza tribe in Tanzania, in the tribes in Papua New Guinea, when the hunters go out and they catch their prey, like in, I think it's in the one in Tanzania, the porcupine is like a delicacy for them. And they're not doing this often. Even though they're called hunter-gatherers, they hunt way less than they gather. They gather a lot more, right? So, but they do go out as hunting parties. One of the first things they do when they capture the animal is they cut open the intestines and then they eat the intestinal content, right? Most of these animals that they eat are ruminant to a certain degree. They're picking up seeds and nuts and all that from all over the place. They're fermenting it in their guts. And if you eat that intestinal content, you're getting all kinds of amazing carbohydrates and resistant starches and so on. So you likely can. If you're eating connective tissue and you're getting different forms of collagen, you're getting some of the glycoproteins from connective tissue. If you're eating organ meat, if you're eating brain, if you're eating liver, you can get diverse enough macronutrient to maintain diversity within the diet. But the unfortunate thing is in the US, when someone's saying they eat a carnivore diet, they're just eating a steak. And that's about it. And I've met a lot of people like that. I mean, I get to speak at places like the Paleo Effects, like I spoke last year. I met so many people at that show that go, yeah, I've gone full carnivore. I'm like, okay, what do you eat? I eat 2 steaks a day. That's it. And sometimes I'll have a dollop of butter on the steak. And I'm like, okay, well, that's not a good thing necessarily. And also that it depends on your microbiome in large part, because if you look at it, one of the ill effects of meat metabolism is the formation of TMAO, right? And there are certain types of bacteria that produce high levels of TMAO. If you happen to be one of those people that have high TMAO producing bacteria and you eat lots and lots of meat, it's going to cause heart disease. There's no ifs, ands, or buts about it. The studies on TMAO are pretty clear. But if you happen to have very low levels of those bacteria, you can sustain this for a period of time until those bacteria grow because you're giving it so much meat, right? So that's another kind of catch to that whole thing, is what happens to the food when it enters your system.
I know this is kind of going off our topic, but I'd love to get your take on this TMAO thing, because those of us who are working with patients with cardiovascular risk factors, we have found that certain supplements like L-carnitine, incredibly helpful for mitochondria, for the strengthening the heart. and choline, super important nutrient for liver health, for brain health, and it's really hard to. It doesn't really seem to make sense. It doesn't fit with our experience that patients who are eating foods or taking supplements with choline or L-carnitine are really putting themselves at increased risk of heart disease.
Yeah, because of the potential
TMAO. So I think
formation of TMAO.
Yeah. So I think the answer to the TMAO question is more about making sure we have a healthy microbiome than about not consuming L-carnitine or choline.
Exactly, yeah, because again, it comes back to that balance issue, right? So one of the things that we test in the Biome Effects test is TMAO producing bacteria. So if
by the way, if everybody's not familiar with this, why don't you explain that Microbiome Labs is offering a stool test, and how can practitioners find out about this?
Yeah, absolutely. So the test is called Biome FX, like the letter F and the letter X. It's a whole genome sequencing test. Just to give you a little bit about the difference between what we're doing and what you have already experienced in the market. We started seeing a lot of issues with accuracy in stool testing. One of the big problems is most of the stool tests, in fact, all of the ones you've used, use something called 16S sequencing. 16S sequencing, if you're not familiar with sequencing information, 16S is like taking random Polaroids of a singular parts of someone's body and then trying to piece it together and identify who that person is, right? So it's a really low resolution, inaccurate way of identifying species level for the bacteria in your system. So it tends to have a very high error rate. And what the companies aren't telling you when you look at your test results and they're showing certain bacteria that they've picked up, they're not telling you that they have an algorithm in their bioinformatics pipeline that makes a prediction that this is likely the bacteria we found. It's not absolute at all. In fact, all of the big microbiome research organizations, the American Gut Project, the Human Microbiome Project, have come out and said you cannot use 16S to identify bacteria to the species level. So we saw that as a big problem, and so we wanted to make that in itself a change. So we started working with the top lab in this space called Cosmos ID, on developing a whole genome sequencing test, which means that we actually have to sequence the bacteria's entire genome from end to end in order to identify it. We're not looking at just little snippets of the bacteria. So it's a far more accurate. It's 99% or more accurate. So you're getting the most accurate species level information. Now, why is species level information so important? Well, because like we've been talking about with the microbiome, it's really about what does that total ecosystem look like, right? You can't necessarily just, outside of the keystone strains, you can't necessarily just pinpoint singular bacteria and their levels as being problematic, because everybody at the species level has most of those bacteria at different frequencies. And so what we look for is really important trends, well-established trends, because in the microbiome, one of the things I say is it's not who's there, it's who else is there, right? The function of certain bacteria are dictated by what other bacteria are in that environment and what are all of their relative abundances. That's where the mapping becomes really important, right? So we can map out functionalities within your microbiome by understanding the species level relative abundance data. And the best researchers who have done this is led by Rita Caldwell. Rita Caldwell is the most decorated scientist in this whole world of microbiome mapping. She's got 60 honorary degrees, including a couple PhDs that she first earned, and then 60 other degrees. She's published 800 papers in this space in peer-reviewed journals, which is a mind-boggling amount for any researcher doing work. So
isn't it the case that the PCR, quantitative PCR testing is actually more sensitive and specific for picking out organisms? And, if you go into a hospital and they suspect C. diff, they're not doing a whole genome sequencing stool test. They're doing a PCR test, right?
They're doing a PCR test, and that's important because the relative abundance of that pathogen compared to all of the other DNA may be really low, right? So for them, in order to find that bacterial DNA or viral DNA in the case of COVID or any other virus, they need to amplify that bacterial virus's DNA, and that's what PCR does, right? The polymerase chain reaction amplifies the DNA. The problem with doing that in a stool test to try to understand your microbiome is it artificially amplifies the volume of that particular species DNA. So it gives you erroneous relative abundance numbers, right? So we need to be able to get really accurate relative abundance numbers. And the thing is that the sequencing work takes longer. That's another reason why if you're ill with something, they're trying to figure out what is making you ill. They use PCR because it can be done pretty quickly. You can get PCR done in an hour or 35, 40 minutes. The genome mapping using full metagenomics takes much longer than that.
Sure.
However, Cosmos ID, the lab that we're working with, are the only lab that are FDA approved to do whole genome sequencing for pathogen identification. Because now we're starting to see some issues with PCR, even in pathogen identification, because you might go to a doc, or you might have diarrhea, and your doctor, your infectious disease doctor, or gastroenterologist might say, you might have C. diff. This looks like C. diff, so let's take a stool sample and then we'll look for C. diff. But they were using the PCR, they will amplify the C. diff DNA and they'll pick it up. Now, C. diff may not even be causing you the problem, right? Because C. diff may be there, but its levels may be low enough that it's not the one causing you the problem. And the PCR is absolutely focused on that one organism. It's not like it's amplifying everything to pick up whatever it can find. It's going in, honing in on that one organism, saying, hey, we found some C. diff, that must be your problem. So we're going to give you vancomycin all day long. Well, that might not be the solution, right? So now the FDA and infectious disease specialists are starting to see that PCR may be problematic even in trying to identify an infectious agent. What may be better is sequencing everything that's there and seeing what is just popped up at the highest that could cause those symptoms. So Cosmos ID is the only FDA-approved lab in the country that can do that kind of work. So we partnered with them to get the best sequencing data. And then our other thing that we focus on the stool test is we want to give you functionality. We want you to be able to tell what your patient's microbiome tends to do and tends not to do, right? It's not absolutes. There's no absolutes within the microbiome, but you will have a guide for you to understand where the real issues may be within that patient's microbiome that you can start honing in on to help. And everything we test actually gives you actionable steps that have references on lifestyle changes, diet changes, and even supplement changes that can help that particular area. Coming back to TMAO, that's one of the things we test, right? So you could eat meat and choline and L-carnitine all the time and be absolutely fine, and it'd be very healthy for you, as long as you don't have really overgrown levels of bacteria that create TMAO. Right? And you may have that because of previous dietary choices, right? When you bring back balance to the microbiome, then you don't have that issue. And so one of the things you can do is you could test your patient's microbiome and say, hey, your TMAO levels are really producing bacteria really high. So let's reduce these kind of things for a period of time until that comes down. Then we can bring them back into the system. Just another example of that, sulfate reducing bacteria, right? Nobody would have really thought about it or heard of these things within functional medicine, but they're so important because one of the things that they do is they take sulfate from sulfate-rich foods and they convert it to hydrogen sulfide in the body, which then becomes really inflammatory to the bowels. And so many healthy foods are high in sulfates. Seafood is high in sulfates, leeks and garlic and artichokes, all of these things, right? High in sulfate. So you might have a patient that's going, doc, I'm eating pretty clean. I'm eating healthy fish, line-caught fish. I'm eating these vegetables. I still have really bad diarrhea. I have all these issues.
I just had a patient today, and she had a flatline SIBO-BREP test, which is usually indicative of hydrogen sulfide.
Yep. Exactly. And so, yeah, and then you will know from the biome effects test, like, oh, okay, it's because your sulfate reducing bacteria are really high. These categories of foods really are actually counterproductive in your gut. So let's bring those down over time, and you could see all of the recommendations that allow to bring that down over time, and then rebalance microbiome. Then you can reintroduce those foods like normal.
So how do we order this stool test, and how much does it cost?
Yeah, so the practitioners, you can order, I think we sell them in bundles, kits of 4. There's no cost to order them, of course, because you can order the kits and have it within your practice until you have the right patient to use it on. The price that we charge is $299. We leave it up to you if you want to mark it up above that, but that's a price that we only talk to practitioners about. So the patients don't know what the cost of this is. We have a mix of practitioners, some that don't mark up tests at all, but then charge a consultation fee to go through it, or some that double the price, depending on how they're set up. So we allow you to decide how to do that. So then the functionality of it is, if you have patients coming into your clinic, you can hand them a test. You can either have you or your staff register the kit for them. It's a very quick process, maybe takes 2 minutes, and you can either put in the payment and then charge them back, or you can have them take the kit home and have them register it using your code in there, and then they will pay for it directly too. So you can manage it however you want. They do this test at home. We've got a very painless sampling procedure. That was another thing that we really wanted to improve. We didn't want people like scooping poop and doing all kinds of stuff. So we have this piece of paper that actually sticks to the toilet seat that has a bowl in it. And then you take your first void, and then we give you a brush, a coring brush, and you kind of core through a few spots in the stool, stick the coring brush in the test tube, close it off, then that's what you mail back. The paper, you can actually just unstick it and flush it. So you're not dealing with actually, managing anything.
That's cool. It's always tricky trying to hold that little French fry basket.
Yeah, exactly. Yeah. And the brush, what's cool about the brush is we even thought about this, like we don't want you to get too close to the stool. So the brush actually has a long handle when you first get it, and you can actually core through the poop sample. I think we recommend somewhere around like 5 or 6 times you core through it. And we use a brush because one of the things we found was you're not getting adequate sampling of the DNA from the stool when you swab the surface or when you scoop a tiny bit of it, right? The DNA and the bacteria in the stool is not homogeneous. So the bacteria on one side is going to look different than the bacteria on the other side, and the relative abundances can change. So we wanted you to be able to go through the whole core of the stool and use a brush with lots of bristles to pick up as much as we can. The more you pick up, the more accurate the data will be. So the brush has a long handle. You core through it a few times. You put it into the test tube, and I think you just break off the top of the handle and then screw the cap on, and then you're just mailing it back in a prepaid envelope. And here's the exciting part about it is we now have an FDA-approved COVID diagnostic in the stool test, right? And it's an actual diagnostic, meaning you can diagnose COVID in the stool. And that's really important because studies have shown that you can actually pick up COVID in the stool for up to 2 to 3 weeks longer than you can pick it up in the upper respiratory tract, right? So you might have a patient that was feeling pretty crummy. And in fact, like we just had this with one of our employees. She was feeling pretty crummy, and she went and got the nasopharyngeal test, and it was negative, right? But there's studies that show you can get up to 50% false negatives with that test. And so she's like sitting there at home having lots of similar COVID-like symptoms and doesn't know if it's COVID or not, right? So we're having her do the stool test instead, and the stool test will come back in about 4 days for the COVID part. The sequencing takes a little bit longer, but the COVID part comes back faster. And if you had somebody that had COVID maybe a couple weeks ago, suspect they did, you could still pick it up in the stool test up to 2-3 weeks after symptoms go away. So it's a great way to do that diagnostic, and you can do it at home. You don't have to go to a testing facility where you're around a bunch of other people that also think they have COVID, and you could kind of keep yourself safe distance from all of that.
This has been an awesome discussion, Kiran. I could talk to you for hours, but
thank you.
Thank you for being so generous with your time. So I think we got to most of people's questions.
Awesome.
And I really appreciate it.
Well, yeah, thank you so much for the opportunity. I always treasure any opportunity to talk about this with people, because clearly everybody here are the people on the front lines. You guys are the ones making the difference out there. And nothing I do is meaningful until it translates to the work that you do. So I'm always grateful for these opportunities. So thank you, Tanya. So great to see you, and thank you for getting this set up for us as well. And really enjoyed it. Really enjoyed it myself.
Excellent. And thanks to everybody.
Yeah. Take care.